All right, so ready to go deeper into the world of cells, beyond those textbooks, huh?
Always ready for that.
Well, get this, we're diving into a paper that really kind of throws a wrench in how we think about cells.
A good one.
It's groundbreaking stuff, but surprisingly, a lot of people haven't heard about it.
Definitely fascinating stuff. We're basically talking about challenging a whole century of thinking about cells and
Yeah,
offering a totally different model.
exactly. It's a 2011 paper by Gilbert Ling
Okay.
called Truth in Basic Biomedical Science Will Set Future Mankind Free. Pretty bold title, huh?
Yeah.
And it lives up to it.
Oh, yeah.
It argues that the typical, membrane theory,
Right.
the one we all probably learned in high school bio,
Yeah.
is actually outdated and maybe even disproven.
That's right. The membrane theory basically says that cells are like these little water balloons
Right.
with a membrane acting as a gatekeeper,
Right.
controlling what goes in and out.
Okay.
Ling argues that it's not as simple as that.
So it's not just this passive barrier. There's more to it.
Much more. To really understand Ling's challenge, we need to go back a bit
Okay.
to 1911 when a scientist named Frederick Domin proposed his theory of membrane equilibrium.
Okay.
This built upon the membrane theory and was the accepted truth for decades.
Wow.
But Ling argues that it has some major flaws.
Okay, so remind me, how was Donan's theory supposed to work?
Well, Donin suggested that the distribution of ions inside and outside the cell was mainly determined by the membrane's permeability
Uh-huh.
and electrical charges.
So if an ion could pass through the membrane, it would distribute evenly.
Exactly, but Ling points to evidence that contradicts this.
Okay.
For example, studies using radioactive tracers showed that substances thought to be impermeable could actually move across the membrane.
Wait, so you're telling me everything we learned in school about this is wrong?
Well, it's not that straightforward. The memory theory isn't entirely wrong, but it might be incomplete.
Okay.
Ling's argument is that it doesn't accurately reflect the complexity of what's actually happening inside the cell. He also points out that the distribution of ions within cells doesn't match up with Donin's predictions.
So the theory doesn't line up with what scientists are actually seeing in the lab.
Right, and on top of that, Ling found a fundamental flaw in Donin's theory itself.
Okay.
It seems to violate the law of macroscopic electroneutrality.
What is that?
To put it simply, this law says that solutions can't have a large imbalance of electrical charge. It's like trying to balance a scale with a ton of weight on one side and nothing on the other. It just doesn't work.
I see. Okay. So I'm starting to see why Lin calls for a major rethink.
Yeah.
So if the membrane theory isn't the whole story, what does he propose instead?
Ling offers a completely different model called the Association-Induction hypothesis, or AI hypothesis for short.
Okay.
And it's based on the idea that the cell's interior is much more structured and organized than previously thought.
Hold on. So it's not just a random jumble of stuff floating around in there.
Not at all. Ling describes the cell's interior as being highly organized at a nanoscale level.
Wow.
He uses the term nano-protoplasm to describe this intricate structure.
So instead of a plain water balloon, it's more like, what, a perfectly engineered machine.
That's a good way to think about it.
Wow.
This nanoprotoplasm is made up of proteins, water molecules, and ions, all interacting in very specific and structured ways. Ling emphasizes two key concepts: association and induction.
Okay, break those down for me. What do those actually mean?
Association refers to how molecules inside the cell interact in close contact, creating order and structure.
Okay.
It's like each molecule has its designated place and they all fit together perfectly.
Like a perfectly choreographed dance inside the cell.
Exactly. And induction refers to the electronic influence these molecules have on each other.
Uh-huh.
This dictates how they bind and behave.
Okay.
Imagine each molecule sending out subtle electronic signals influencing the others around it.
So it's not just about the molecules themselves, but also these subtle electronic interactions between them.
That's the core of the AI hypothesis, and this has major implications for how we understand the way cells function, like how they distribute substances, allow things in and out, and even maintain their electrical charge.
Sounds like we're just scratching the surface here.
We are!
Yeah.
The AI hypothesis offers a radically different way of looking at cells, and by extension, at life itself.
I'm definitely hooked. Let's dive deeper into how this model actually explains these crucial cell functions.
Absolutely. Let's start with solute distribution.
Okay.
How cells manage the different substances within them.
Okay, so the membrane theory says it's all about what can and can't pass through the membrane.
Right.
What does the AI hypothesis say?
Ling argues that it's more about selective absorption instead of just floating around freely Exactly. Ions like potassium are actually bound to specific sites within the cell.
So the cell is more like a carefully organized library where each ion has its designated shelf,
Right.
instead of just a bag where everything mixes randomly.
That's a great analogy. This explains why certain ions are concentrated in cells, even though they could theoretically pass through the membrane.
Okay.
It's not just about the membrane, it's about the internal structure of the cell and how it actively manages its contents.
Wow, that's much more dynamic than what I learned in school. So how does the AI hypothesis explain permeability, the way things get in and out of the cell?
Ling suggests there are multiple pathways involved,
Gotcha.
not just simple diffusion through the membrane. Some substances might move through water channels,
Right?
while others bind and unbind from cellular components.
So it's like the cell has a whole security system, not just a single door.
Exactly, and this explains how cells can control the permeability of the same substance under different conditions,
Huh.
something the traditional view struggles to explain.
It's almost like the cell can choose what comes in and goes out, depending on its needs.
Right, and then there's the cellular electric potential. Remember, the traditional view is that this resting potential is just a difference in charge across the cell membrane.
Right, like a battery with a positive and negative terminal.
But Ling proposes that the resting potential actually comes from charge separation within the structured nanoprotoplasm itself.
Okay?
It's a product of that intricate internal organization we talked about.
So it's more like a subtle hum coming from the entire cell, not just a static charge on the surface.
That's a good way to visualize it.
Okay.
And finally, Ling's model offers a fresh perspective on how cells swell and shrink.
The textbook explanation is all about osmotic pressure, right?
Right.
Water flowing in or out depending on the concentration of solutes.
Right, but Ling suggests that it's actually more about changes in the electronic environment within the cell.
Okay.
These changes affect how water molecules bind and influence the overall stability of the cell's structure.
So swelling isn't just about water rushing in. It's about a more complex interplay between the cell structure, its electronic environment, and the water molecules.
Exactly. He even gives the example of how cells swell differently in potassium chloride versus sodium chloride solutions.
Oh, wow.
Both solutions have the same concentration,
Okay.
but cells swell much more in potassium chloride.
So it's not just about the concentration of the solution, it's about how specific ions interact with the cell's internal structure.
That's exactly what Ling suggests.
Wow.
His AI hypothesis argues that potassium ions, due to their specific electronic properties, disrupt the cell structure in a way that leads to greater water uptake and swelling.
It's amazing how much more there is to this than what I learned in biology class.
It is fascinating.
So basically, Ling is saying that the AI hypothesis explains these four key cell functions: solute distribution, permeability, cellular potential, and swelling in a way that the traditional membrane theory simply can't.
That's a great summary. He's proposing a real paradigm shift in how we understand the very basics of how cells work.
And it's not just that he's presenting an alternative theory, he's actually quite frustrated that the membrane theory still dominates textbooks despite these challenges.
He is, and he makes a passionate call to action,
Right.
especially to young scientists and teachers, urging them to take a fresh look at the evidence and be open to this new way of thinking.
So he's really saying, don't just accept what you've been taught, question everything, look at the evidence, and be open to new possibilities.
Exactly. And that's what makes this deep dive so exciting.
Yeah.
It's challenging us to question even the most fundamental concepts and explore alternative explanations.
I have to admit, I'm already starting to see the world of cells in a whole new light. But I'm curious, if the membrane isn't the main player, how does Ling explain the dynamism of the cell?
Yeah.
How do these nanoprotoplasmic units actually create the phenomena of life?
That's where Ling introduces two fascinating concepts: the living state and the active living state.
Okay, I'm ready for more. Let's unpack those ideas.
Ling argues that a living, functioning nanoprotoplasmic unit exists in a specific configuration. He calls it a fully extended state.
So picture a chain of molecules all stretched out, ready to work.
Yeah, that's a good way to visualize it. In this state, all the components, the proteins, water molecules, and ions are closely associated and interacting with each other in a very ordered way.
And what keeps this living state going?
That's where ATP comes in, adenosine triphosphate, the energy currency of the cell.
Yeah, I remember that from bio. It's involved in basically everything cells do.
Exactly. Ling argues that ATP is essential for keeping the nanoprotoplasm in this fully extended, living state. He calls it an electron withdrawing cardinal absorbent, or EWC for short.
EWC catchy. But in simple terms, what does that actually mean?
Think of it like this. ATP acts like a tiny magnet, helping to hold this whole delicate structure together in its active, life-sustaining configuration.
Okay, I think I'm getting that. So the cell needs a constant supply of ATP to maintain this living state. But how does it actually create action?
Right.
Cells aren't static. They're constantly doing things,
Right.
responding to their environment.
That's where the active living state comes in, when a nanoprotoplasmic unit is stimulated, say, by a nerve impulse or a change in its environment. It momentarily shifts out of its fully extended state.
So the chain of molecules kind of folds up for a bit.
Exactly. And this folding changes how the ions and water molecules are arranged. It also releases some ATP.
So it's like a controlled collapse and re-expansion, and that's what drives cellular activity.
Yeah,
It's kind of like a muscle flexing and then relaxing again.
that's a great analogy. In fact, Ling uses muscle contraction as an example to explain this. When a muscle is relaxed, its
Okay.
nanoprotoplasm is in the fully extended living state, but when it's stimulated to
It
contract,
shifts to this active living state.
exactly, ATP is released, the protein chains fold up ions, and water molecules rearrange,
Wow.
and the muscle fibers shorten, causing the contraction.
Okay, that makes sense. But then how does the muscle relax again?
Once the stimulation is over, the cell replenishes its ATP, the protein chains extend back out, everything goes back to its original arrangement,
Uh-huh.
and the muscle relaxes.
It's like a perfectly coordinated dance, driven by ATP and influenced by what's happening around the cell.
That's a beautiful way to put it.
Wow.
And Ling argues that this back and forth between the living state and the active living state is basically what life is all about, this constant dynamic interplay.
So what happens if the cell can't get enough ATP to maintain this dance?
Ling says that if a cell is deprived of ATP for too long, it can't go back to its fully extended state.
Huh.
It gets stuck in a permanently contracted state. Imagine those muscle fibers staying permanently clenched.
That sounds not good.
It's not. That's essentially what we call cell death. The nanoprotoplasm loses its organized structure and can no longer shift between states. The cell can't function anymore.
Wow. Okay. So life, according to Ling, is this constant dance between these two states,
Right.
fueled by ATP, and death is when the music stops.
That's the central ideal, and he argues that this model, based on association and induction,
Right?
actually explains the phenomena of life better than the traditional membrane-focused view.
I'm starting to see how elegant this model is. It's not just about individual molecules or parts. It's about how they all work together in this dynamic system. But there's still one thing I'm not quite getting.
Okay.
How do specific molecules know where to go and what to bind to in this whole dance?
Yeah.
How does the cell orchestrate all of this with such precision?
That's where things get even more interesting. Ling introduces two key ideas called C-value and C-value analog.
Here we go, more new terms, but I'm ready. What are these C values all about?
Think of C-values like electronic signatures. Every molecule and every binding site within the nanoprotoplasm has a specific C-value.
So it's like a unique electronic ID card that dictates how each molecule interacts with others.
Exactly. The C-value determines how strongly a site will attract a particular ion or molecule.
So a site with a low C value might be really into potassium ions,
Right.
while one with a high C value might prefer sodium ions.
That's a good way to put it. And what's even cooler is that these C-values aren't fixed.
Really?
The cell can actually change them.
Wait, really? So the cell can fine-tune these interactions by adjusting the C values?
Yeah.
That's like having a control panel for molecular attraction.
That's a great analogy. And Ling believes that this ability to modulate C-values is a key mechanism for regulating cellular activity.
So it's not just about the amount of ATP, it's also about these subtle electronic interactions controlled by C values that determine how the cell functions, how life happens.
You've got it.
Wow.
It's a complex model, but it paints a beautiful picture of the cell as a dynamic, self-regulating system.
This is mind-blowing. I'm already starting to think about the implications. If cells are this intricate and self-organized, what does that mean for our understanding of health and disease?
Absolutely.
I mean, it completely changes how we think about what causes illness, doesn't it?
Absolutely. And Ling dives into those very questions. He argues that the AI hypothesis could revolutionize our approach to medicine. But we're running out of time for this part of our deep dive.
I guess we'll have to save those revolutionary ideas for next time. I'm definitely on the edge of my seat. Welcome back to our deep dive into Gilbert Ling's fascinating AI hypothesis. And let's be honest, pretty mind-bending.
It is.
We've explored how it challenges conventional cell biology.
Right.
But now I'm really curious how Ling applies this to, like, our understanding of health and disease.
Well, one of the biggest differences is in how we view the very causes of disease.
Okay.
The typical medical model often focuses on things like bacteria, viruses, or genetic mutations.
Right. It's either something invading from the outside or a glitch in our DNA.
Exactly. But Ling argues that this focus on external or genetic factors might miss a crucial piece of the puzzle, the cell's internal environment.
Uh-huh.
Remember, the AI hypothesis puts that internal environment front and center.
So it's not just about finding the bad guy, but understanding the terrain where the battle's being fought.
That's a great way to put it. Ling suggests that many diseases, even those with a genetic component, ultimately come down to disruptions in the delicate balance within the cell's nanoprotoplasm, that balance of association and induction that we talked about.
So even if you have a faulty gene, the real problem might be how that gene, or maybe some environmental factor, messes up the intricate workings inside the cell.
Precisely. And this shift in perspective could change how we approach treatment.
Because if the root of the problem is in the cell's internal environment,
Yeah,
then just focusing on killing a virus or fixing a gene might not be enough.
that's what Ling is saying. He believes a more effective approach would focus on restoring the proper balance within the cell, essentially, creating a healthier terrain.
It's like instead of just attacking the enemy, we're also making sure our defenses are strong and the environment is inhospitable to disease.
Exactly. And this idea that the cell's internal environment is so crucial to health really resonates with a growing movement in medicine called functional medicine.
Oh yeah, I've heard that term before. It's more about a holistic approach, right? Not just treating symptoms, but looking for underlying imbalances.
That's it. Functional medicine practitioners try to pinpoint and address the root causes of disease,
Yeah.
looking at things like nutrition, lifestyle, environmental exposures, all factors that can affect that cellular environment.
And Ling's AI hypothesis seems to provide a solid scientific basis for understanding how those factors can impact things at the cellular level.
It does. It's a model that connects the microscopic world of molecules to the bigger picture of health and disease.
Like Ling is offering a whole new lens for looking at human health, one that emphasizes this intricate web of interactions within our cells and the importance of that balanced internal environment.
You've summed it up beautifully, and it doesn't stop there.
Okay?
Ling even suggests that the AI hypothesis might shed light on something as complex as consciousness.
Wait, hold on. We're going from cells to consciousness now.
We are.
That's a big leap.
It is, but Ling argues that if the cell, the basic unit of life, is this incredibly intricate, self-regulating system, maybe consciousness itself arises from a similar principle operating at a higher level.
So instead of consciousness being some separate, mysterious thing,
Yeah,
maybe it's woven into the very fabric of how matter organizes itself.
that's the idea. He's suggesting that consciousness might not be magical or inexplicable, but a natural outcome of the way complex systems, like our brains, are organized.
So consciousness isn't out there somewhere. It's built into the universe, a product of matter itself.
It's a profound thought, isn't it?
Yeah,
And while it's certainly speculative, it shows just how expansive Ling's thinking is.
right.
He's willing to challenge those conventional boundaries and explore connections between seemingly unrelated areas.
We've gone from the inner workings of a single cell to rethinking health and disease.
Yeah.
And now we're pondering the nature of consciousness. This deep dive has really stretched my brain.
It certainly has been a journey. It shows the power of science to question, to push boundaries, and to open up whole new ways of seeing the world, both around us and within us.
Before we wrap up, is there one final, thought-provoking question you think our listeners should ponder? Something that builds on Ling's work, but that we didn't get to directly.
I think a particularly interesting question is this. If the cell is this self-organizing system, what does that tell us about evolution?
Because the traditional view is all about random mutations and natural selection, a process often seen as blind and purposeless.
Right. But if the cell itself is inherently organized and responsive to its environment,
Yeah.
could evolution be more directed, more influenced by how organisms interact with their surroundings?
So instead of just passively waiting for random genetic changes, organisms might actually be active participants in shaping their own evolution.
That's the question Ling's work raises. It challenges us to rethink our assumptions about life, evolution, and our place in the grand scheme of things.
That's a perfect note to end on, a reminder that the search for understanding is never truly finished, and even the most basic ideas can be challenged and reimagined.
It's been a pleasure diving deep with you today.
And a huge thank you to you, our listener, for joining us on this incredible intellectual adventure. We hope this has sparked your curiosity and that you'll continue to question, explore, and push the boundaries of your own understanding. Until next time, 70 time.